Hello students. So we are going to continue our discussion on enzymes. Okay. So we have stopped at factors affecting enzyme activity. Now we are going to start with inhibition of enzymes. Okay. We'll now start with inhibition of enzymes. Okay. Now the type of inhibition that is given in your NCRT, this is known as what? It is known as competitive inhibition. Okay. Competitive inhibition is the type of inhibition which is given in your NCRT. Now, suppose you have an enzyme. Suppose this is an enzyme here, right? And this enzyme has an active site, right? This is an active site, okay? Now, there are two substrates, fine. Suppose this is substrate A and there is another one which is substrate B, right? Now, if substrate A and B are exactly similar, if substrates A and B are similar in shape, hmm, what will happen then is that A and B may compete for binding in the active site, right? It's like competing for the same position, right? For example, that you're giving an entrance exam, and there is only one seat left. So A and B are the two candidates, right? They're aiming for the same seat. So will they not compete with each other, right? So if suppose this is meant to be the substrate, okay, if suppose this is the original substrate, and if B competes with this A to bind to this active site, then B basically inhibits the binding of A in a way, right? So B becomes the inhibitor, okay? B then becomes the inhibitor. Now this is what essentially competitive inhibition means. Now if we look at the structures of competitive inhibition, right? Let me just go straight down to the structures. Okay. See, competitive inhibitor is basically it is a substrate which closely resemble the substrate in its molecular structure, right? So if they are similar in shape, they can inhibit the activity of the enzyme. Okay. Why? Because if the original substrate, for example, suppose the original substrate is A and B is the inhibitor, what is going to happen is that B is going to compete for binding. So basically, both of them are competing for the same spot, right? So this is inadvertently going to lessen the number of bindings of A, right? Because there won't be as much A molecules binding to the enzyme. Is this point clear? Now, uh, see this reaction, this entire thing, competitive inhibition is completely reversible. Okay, competitive inhibition is completely reversible. It is a reversible inhibition. Okay, it is a reversible inhibition. How do you reverse this inhibition? The only thing that you have to do is increase the number of substrate. Just a second. The only thing that you have to do is increase the number of substrates, right? Okay, in this case, you have to increase the number of A. Fine. So this way, competitive inhibition can be reversed. Now, after competitive inhibition, there is the classification of enzymes. So enzymes can be classified into six categories. Okay, they can be classified into six categories. Let me just elaborate on them a little bit. Okay, the first category is oxidoreductase. Okay, so Oxidoreductase are basically enzymes which can undergo redox reactions. Okay, I hope you know what are redox reactions, right? So if one substrate is reduced, the other will be oxidized. Okay, transferases basically means that you transfer a certain group from one substrate to another. For example, this entity G, okay, if you look at it carefully, in the reactant side G was with S, and in the product side G is with S naught, right? Fine. So basically G has been transferred from S to S naught, right? Enzymes which helps in catalyzing transfer of certain groups, they are known as transferases. Then comes hydrolases. Hydrolases basically means enzymes which can break down, okay, or undergo hydrolysis. Okay, so there are different bonds in different bonds in biology, like ester bonds, ether bonds, peptide bonds, glycosidic bonds, carbon-carbon bond, carbon halide bond, phosphorus and nitrogen bond, right? So basically what happens here is that hydrolysis, the word hydrolysis basically means breakdown by water. Okay, breakdown by water. Hydro means water, lysis means basically breakdown. So if a molecule or a bond is broken down by water, enzymes which helps in this breakdown, they are known as hydrolysis. Okay, hydrolases for it. Then comes lyases. Lyases basically means breaking down. Lyase also means breakdown, but without water. Okay, basically breakdown without water. All right. So if you see here, these two bonds, CX and CY bonds were broken down. So there's a C double bond, C bond formed, and XY forms a separate bond altogether. So basically, lyases also means basically breaking down, and hydrolases also means breaking down. But lyases are basically enzymes which helps in the breakdown without the help of water. Hydrolases are enzymes which helps in the breakdown of organic covalent bonds with the help of water. Okay. Then comes isomerases. Okay. Isomerases converts a molecule to its isomer. For example, let me just give you an example. Glucose 6 phosphate gets converted to fructose 6 phosphate. Okay. Now, glucose and fructose are isomers, right? And this particular enzyme, okay, this particular enzyme which converts this glucose into fructose. Basically, glucose and fructose are isomers. What are isomers? Isomers are molecules having the same molecular formula but different structural formula. Okay, so such enzymes are known as isomerases. Okay, fine. So if a molecule is converted into an isomer, such the enzymes which help in this conversion is known as isomerases. Then comes ligases. Ligases basically are enzymes used for joining. Okay, they're used for joining, right? So basically, ligases are used for building a bond. Okay, bonds like carbon, oxygen, carbon, sulfur, carbon, nitrogen, phosphorus, oxygen. So in order to build all those bonds or to join all those bonds, you need ligases, right? Now comes cofactors. Okay, now see, uh, enzymes can be of different types. Okay, enzymes can be basically of different types. Now, just give me a minute. Okay. See, enzymes, like we were saying, enzymes can be of two types: simple enzyme and conjugated enzyme. All right. Now, simple enzyme comprise only of proteins. Okay. So, if an enzyme only has proteins, such enzymes are basically known as simple enzymes. Conjugated enzymes basically means that an enzyme will have a protein component. Also, it is also going to have a non-protein component. Okay. It is going to have a protein component and also a non-protein component. Right. Now, if we look at conjugated enzyme properly, hmm, conjugated enzyme, okay, the protein component of conjugated enzyme is known as apoenzyme. It's known as what? Apoenzyme. This is the protein component. And the non-protein component is known as cofactor. This is known as what? Cofactor. Is this point clear? Okay. So, apoenzyme and cofactor together makes hollow enzyme. Okay. It makes hollow enzyme. This is very important. Now, basically what happens is that cofactors can be of different types, right? So, let me, okay. Cofactors can be of different types. So, let me just elaborate it here. Okay. So, I think you have already seen the basic outline. So, let me just write what are cofactors again. Hmm. See, cofactors can be of different types. Okay. Uh, if the cofactor is organic, hmm, then there can be of two types. Okay. If the organic cofactor is easily separated, then this is known as a coenzyme. If the organic cofactor is not easily separated, okay, if it is not easily separated, then it is known as a prosthetic group. This is known as prosthetic group. And the cofactor also can be inorganic metallic ions. Okay, it can also be inorganic, and inorganic means basically metallic ions. So these are basically activators. These are all activators. So this is the basic classification of an enzyme. All right. I hope this video was useful. You can pause the video, rewind the video wherever there is doubt, wherever there is query. I hope you found it useful. Thank you so much.